I am delighted to be joined by Mike Severino, the CEO of Tessera Therapeutics, at our flagship studio today. And Mike comes to San Francisco with strong data in hand and a freshly inked partnership. So Mike, let's start there. Let's start with sickle cell disease. Tell me a little bit about the data that was presented this year and how your in vivo approach is different than others who are also looking at sickle cell disease.
Well, thanks, Lindy. It's a pleasure to be here. And I'm really excited about the progress we're making in sickle cell disease and a number of areas in our pipeline. Sickle cell disease, I think people are broadly familiar with, but one of the things that I think people don't realize is that there are about 250,000 patients in the developed world struggling with sickle cell disease. There are about 6 million patients globally. And while there have been recent advances in therapy for sickle cell disease, there are no curative approaches that are broadly applicable. Any curative approach today requires a stem cell transplant to deliver. So that means a patient has to have their stem cells collected, modified outside of their body. And then, even more importantly, to re-administer those cells, they have to get high doses of toxic chemotherapy to completely destroy their pre-existing marrow so that they can be rescued.
Yeah.
It's great to cure any patient of sickle cell disease, but an approach like that is never going to be scalable. It's not going to be scalable in the developed world, and it's going to be even more challenging in underserved regions. That's not the approach we're taking. We've developed two very important technology platforms. One, we have developed the ability to rewrite the sickle mutation back to the intended sequence, so back to its normal, healthy hemoglobin adult A form. And that's absolutely essential, but just as important is the ability to deliver it in vivo. What that means is we can take a simple IV infusion and target lipid nanoparticles to take that gene-writing construct directly to the stem cells in the marrow in their native niche without any other manipulation, without the need to mobilize them, without any off-site manufacturing, and most importantly, without chemotherapy conditioning. We're really, really excited about this approach, and at ASH we just showed that in humanized mice, we can correct with 62% efficiency the hemoglobin locus. And that is well above the curative threshold, which is anticipated to only be about 20%. So those data, I think, really caught a lot of people's attention. And we're very excited to move this toward the clinic because we think this will be the first sickle cell program with curative intent that can be broadly applied in the developed world, and something that can be helpful to patients in underserved regions as well.
Incredible technology advancements, particularly over the last year. And that data at ASH, data presented earlier last year, captured the attention of the Gates Foundation. So tell us a little bit about that.
Absolutely. So the Gates Foundation has obviously done a tremendous amount of work in underserved regions and is dedicated to bringing novel therapeutics around the world where they can help people who are wrestling with life-threatening and unfortunately life-shortening illnesses. And the sickle cell data certainly caught their attention. We've had a number of conversations with the Gates Foundation. And they share our view that this is the kind of therapy that we can take around the world. I said there are 250,000 patients in the developed world. There are 6 million patients globally. Many of those live in equatorial Africa or other regions around the equator. In India, there's a large focus as well around the equator. And many of these patients are in underserved regions and just, quite simply could never afford a stem cell transplant or would never be able to have one offered to them. This kind of therapy can really be, in our opinion, a lifeline for those patients. And so we have committed with the Gates Foundation to making this therapy available once it's approved around the world, including in those underserved regions.
Mike, we're here at JP Morgan. A big focus of the conference is around partnerships, and knowing that those discussions might start here and take months or perhaps even a year to come to fruition. Tell us a little bit about how the relationship with the Gates Foundation started.
Well, it started with the data. They had seen us present at meetings, reached out to us. They have a tremendous amount of expertise in this area. The person who leads that area is a very well-known and very well-established stem cell biologist. And it really started with conversations around the data. The sorts of results we have achieved in terms of delivering to long-term stem cells in the marrow just weren't supposed to be possible. Nobody thought it was possible. We didn't know it was possible until we started on this journey. But we've achieved remarkable results, and that caught their attention.
Right.
They watched it develop, and we decided to do this partnership together right around the end of last year.
Very exciting. So speaking of figuring out what's possible and learning what's possible, you were also focused on AATD, which is a disease that maybe people haven't heard as much as they have for sickle cell. Help us understand the optimal target product profile that you would be bringing forward as a gene writer solution for that disease.
Certainly. Alpha-1 antitrypsin deficiency in some ways is the most common life-shortening genetic illness that many people have never heard of. And the reason I think that many people haven't heard of it is it looks like other things. It looks a bit like chronic obstructive pulmonary disease, but it's much more rapidly progressive. And it looks like liver disease. It can have other causes. So it's often underdiagnosed. But there are between about 200 and 250,000 patients in the US and Europe who have the single mutation that causes alpha-1 antitrypsin deficiency. So what that mutation does is it alters a protein that's formed in the liver, and it makes an abnormal form that has two consequences. That abnormal form sticks to itself and forms aggregates. And because it sticks to itself, it can't get out of the liver, and therefore it can't do its job. And the job of this protein is to protect the lungs from destruction by our immune systems in the course of immune surveillance that occurs every day of our lives. So that's where the lung disease comes from, and unfortunately the lung disease is the most common cause of death in these patients, and so it's really quite serious, quite severe. But there's a second problem. Because those aggregates form in the liver, they're directly toxic to liver cells, and so these patients get, many of them, an advanced liver disease, and a third of them go on to have cirrhosis. So the ideal therapy would correct both of those problems. You'd correct that abnormal protein back to its intended sequence. It could get out of the liver, protect the lungs, and you could spare the liver from damage. And we have an agent that shows that sort of activity in a number of different preclinical models, in humanized mouse models. We've also taken it into non-human primates, and we're showing remarkable efficiency. In a humanized mouse that bears the human gene that causes this disease, we can correct between about 93 and 95% of the disease-causing mutation. You only need to correct about 30% to cure this disease, the field thinks. So we're well above that level. And that should protect the lungs from destruction, and it should spare the liver disease as well. So it's another program that we're very excited about.
Really neat. And in both cases with sickle cell and AATD, you're correcting a single mutation back to the original.
Yeah, that's what's the most exciting thing about our technology. For the entire history of medicine, we've tried to look for ways to sort of work around a problem, to find a lever that can maybe make symptoms a little bit better, slow progression down a bit if we can. And if that's all you can do, it's a worthwhile endeavor. But really, for the first time in medicine, we can go after the causative mutation and correct it back to wild type.
Incredible.
Wild type means the sequence that healthy individuals around the world have, the intended sequence. And by doing that, we can actually think about cure on a scale that's just never been possible in medicine.
So, Mike, you have a pipeline that's starting to emerge, an investment from a global NGO. What else is in store for Tessera in 2025?
Well, there's a lot that's in store. We're going to move the programs we talked about and more forward. Alpha-1 antitrypsin deficiency, a program that we didn't even have a chance to talk about called PKU, and metabolic disorders and sickle cell disease are all moving rapidly towards the clinic. We're already building the base for our next important wave of programs. That'll be our second wave in the clinic, if you will. Areas like CAR T generation in vivo, so without offsite manufacturing, without chemotherapy conditioning, for either cancer or autoimmunity, and much, much more. So it's going to be an exciting 2025 for us.
Just keep building on and on and on from the progress that you keep making each year. It was really exciting, Mike. Thank you so much for sharing with us and for being here.
It's a pleasure to talk to you. Thank you.